Thanks, Kevin and Daria. And I also want to say when I give talks around the country and people want to know how we did what we did, and I tell them about SPARK, I think people are really envious. And I know a lot of major institutions are trying to model SPARK, and they're trying, but I don't think they've got there yet. So I want to acknowledge their contribution up front. What I'll be talking about today is the development of Ubenamx, which is an LTB4 inhibitor, LTB4 being an inflammatory mediator for the treatment of pulmonary hypertension. Etta told us about PH. It's a lethal disease that affects mainly young to middle-aged women. And I'll also talk about how this drug may also be useful in a chronic condition known as lymphedema, which is a problem of third world countries that have parasitic infections, namely filariasis, and also in this country, mainly for the treatment of, or a problem of secondary to mastectomy and lymph node resection in breast cancer patients. So what I'll talk about briefly then is about how LTB4 works to cause pulmonary hypertension, how it works to potentially cause lymphedema, which will be very brief, and then finally we'll conclude talking about SPARK and bringing Ubenamx forward with Eiger Biopharmaceuticals. So just jumping straight to the conclusion of our paper, which we published a couple of years ago, what we think happens in pulmonary hypertension is that there's an initial injury to the vascular endothelium, and this can be caused by a number of things like vasculopathic viruses or autoimmunity. And if you have a non-robust immune regulation response, in other words, you might not have regulatory T cells that quell the inflammation well, you get an overly exuberant inflammatory response. And one of the things that we see in our animal models of disease as well as clinical samples is that the macrophages are particularly activated among all of the other inflamed cells that we see. And what we saw and discovered in this paper is that macrophages secreting this molecule, leukotriene B4, was capable of actually killing the cells in the inner lining as well as growing the smooth muscles on the outside. So these smooth muscles undergo hypertrophy and proliferation, and you also get an expansion of the fibroblast layer on the outside. As Kevin and Etta both mentioned, currently right now this multi-billion-dollar industry of drug companies that target pH are just looking at targeting this pulmonary smooth muscle layer which has inappropriate vasoconstriction. So there's no treatments right now that actually try and reverse the revascular remodeling. And so what we think happens with this disease is that you have an overabundance of leukotriene B4 in some patients. This actually couples with abnormal BMP signaling that Etta was speaking about earlier, and this leads to an occlusion of the vessels. You have a high pressure head, right ventricular failure, and death. So what we found was basically in that paper also is that patients also have high LTB4 levels, and you find very high levels of LTB4 in the BAL fluid of animals that suffer the disease, and targeting that, those pathways work to reverse disease. So just getting back to a little history then about how we worked with SPARK, Jay Rajadas, who directs the BioADD facility and is a formulation chemist, was working with SPARK and helped us in the early period developing inhalable drug. This is an orally bioavailable drug, but we developed multiple formulations trying to figure out what would be the best way to bring this to market. We made the contact with Jay. Jay introduced us to SPARK. We then put in intellectual property in 2011, had our first SPARK grant, and then with the help of Kevin and Daria, met many SPARK advisors, which led us to meeting our patent attorney, Kevin Castor, in 2011, who introduced us to our future CEO. And this is the thing that I think was missing in a lot of the investigators around the country. They didn't have that connection to the advisors and to potential CEOs. And David Corey has a huge amount of industry experience and has been instrumental in bringing us forward. So in September of 2012, we developed a SPARK grant to develop new LTB4 inhibitors. I have to say at the time, there was a lot of confusion because like FK506, this was a repurposed drug, but what we didn't know is that the fact that it was only being used in Japan mattered in terms of a business model. The other thing that was going on right at that time is that I like my job at Stanford. I really want to be a faculty member, and I try not to be compromised with NIH funding. And as we move forward with talking about businesses and profits, it's very important for us to know about conflicts of interest, and so we learned all about that, and Stanford's been outstanding with that. So as this was going on, I think one of the great things about Stanford is that it's relatively small in size and with very high-level people here. And one of those people that was working with Kevin and Daria at the time was an investigator by the name of Stan Roxon, who's chief of the cardiology consult service. And Kevin had heard some information about this condition, lymphedema, on a SPARK project that Stanley was involved with. And it reminded Kevin of a lot of what we were doing in our PAH model. And he said, I don't know, I got this hunch that there might be some connection between what you guys are doing and what Stan's doing. And so Stan didn't have a big lab operation, so he asked me to meet with Stan, and I did. Turned out Stan had this PLOS One paper back in 2009, which showed that ketoprofen was working in preclinical lymphedema. Now, lymphedema, which I'm going to talk about in a sec, is a condition that has no medical therapy. When you have lymphedema, you have it for life. All you can do is wear compression stockings or wraps. There's really nothing that works for it. And Stanley, who's one of the leaders in the country with lymphedema research, realized that inflammation, along with many of his colleagues, that inflammation was likely contributing. And one of the things that we worked with Stan to evaluate was that when ketoprofen works, it works not only by blocking COX-2, but leukotriene B4. The problem with ketoprofen, though, is that it's limited by GI and renal toxicity. So even as Stan was moving forward to clinical trials in humans, so one of the first human trials for for lymphedema, we were starting to wondering maybe there's a better pathway for you to target that doesn't have these toxicities. So lymphedema is a disease basically that evolves when you have impaired lymphatic circulation. So normally the lymphatics drain fluids from your extremities. If you block that circulation, you have this very dilated, kind of like elephant man-like limbs, and it is very it creates problems for basically continuing your work, and it leads to social isolation. So again, this is a process that we see when you remove the breast and you have lymphatic obstruction. We developed with Stanley a mouse tail model, and what you can see is that when you sever the lymphatics in mice, the tails get very voluminous, and they get somewhat better, but they stay voluminous for life. And what we were able to find was that while ketoprofen did work in this model, blocking LTB4 with a less toxic agent, Ubenimex, was just as effective. And I won't get into the molecular mechanism, but it was just a coincidence, and it really is quite unique from the way that it works in pulmonary hypertension. So moving the basic science then into a new treatment for disease, David Quarry, as we were developing the PH story, flew to Japan. He met with the company that's responsible for the production of Ubenimex in Japan. Ubenimex is a neoadjuvant agent that has very little toxicity. This company does not get itself involved with pharmaceuticals much. They really make tires, iPhone covers, and Ubenimex just by coincidence. And it turns out, which I didn't know at the time, and David was aware, that there is a business model if a repurposed drug is not being used in other parts of the world, that you can turn it into something that works. So the Nippon Kayaku team flew out to us, met with the team, decided they liked us and wanted to do business with us. We finally published the paper, had many VC presentations, and basically founded a company, which was actually ICOS, called ICOS, E-I-C-C-O-S-E, not another spelling. And that was brought into Eiger Biopharmaceuticals, which was founded by Jeff Glenn, sitting in the audience. And this is a company that focuses on orphan diseases. They have a very experienced pharma team. And I want to also acknowledge, beyond David Quarry, Joanne Kwan, who's our chief medical officer, also a Stanford medicine grad. And this led to kind of the more modern incarnation of Eiger Biopharmaceuticals, which is now located in Palo Alto. This formation of the company, they went through a reverse merger with Celadon earlier in the spring, are publicly traded now on NASDAQ, and this allowed us to go forward with 2 clinical trials associated with this, the hepatitis delta program already underway. Liberty is now has 45 centers around the U.S. and Canada, with 25 centers activated, and that's already enrolling patients. The Ultra trial is testing Ubenimex in lymphedema and is already enrolling patients. So to summarize, targeting LTB4 for PH and lymphedema was found to be effective in preclinical studies. SPARKee was instrumental in helping us develop the translational science, and as you might expect in Silicon Valley, the introductions were everything and helping our company get off the ground. I want to acknowledge Kevin especially for making that connection with the distinct biology of lymphedema with our story just on a good hunch. And so now we're moving forward with the trial. Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.